Welcome, everyone. I'm Thomas Sir, CEO of Kinexum. Today's joint Kinexum Cello Health BioConsulting public webcast features some remarks with slides by Dr. Brian Leyland-Jones of Avera Cancer Institute and Dr. Jeff Bockman, Oncology Practice Head at Cello Health BioConsulting, and a fireside chat format conversation on the topic of precision medicine approach to cancer care and prevention, successes and challenges 2019. A couple housekeeping points first. If you have a question along the way or at the conclusion of the presentation, please enter it in the question box that you can open up from the options on the right of your screen, and Jeff will raise as many as we have time for. Next, a recording of this webcast with slides will be posted, and a link will be distributed to registrants of this webcast within the next several business days. And finally, I want to sneak in plugs for a couple future Kinexum events. Our May public webcast will be on the revolutionary changes in drug regulation in China over the past couple years and what that should mean for Western drug developers, featuring George Bader, SVP of DMed, a Shanghai-based CRO founded by former Big Pharma executives and FDA officials. And the second is our Metabesity Conference, October 15 to 16, at the Carnegie Institution for Science in Washington, D.C. You can check out our stellar growing speaker roster, which includes Victor Zhao, President of the National Academy of Medicine, Richard Hodis, Director of the National Institute on Aging, and Janet Woodcock, Director of CDER at FDA. Information on both events can be found on our website, kinexum.com. I'd now like to introduce Dr. Jeff Bockman, EVP and Oncology Practice Head at Cello Health BioConsulting, formerly Defined Health, to start the contents. Jeff?
Thank you, Thomas, and thanks everyone for attending, and a special appreciation to Dr. Brian Leyland-Jones, who got caught in the bomb cyclone and fortunately has made it back home to be able to participate here today. I did want to note that we do have some of our own upcoming events. In particular, we hope people will be planning to attend our 30th annual Cancer Progress event here in New York City, May 7th to 8th. Some great keynote speakers, as you can see, and phenomenal set of panels across various aspects of scientific, clinical, and commercial issues within oncology, including, of course, a number of panels on immuno-oncology. So with I'm going to give a bit of a general comments here to kind of set us up for the discussion, and then turn this over to Brian to give a brief talk about many of the issues that we're facing, some of the successes and remaining challenges as we try to bring precision medicine to cancer and to more and more cancer patients.
So I just want to personally just say good morning to everyone. Thank you for joining, and I promise I will keep my slides down to about 13 minutes.
Great, thank you, Brian. So, you know, what we're basically going to be attempting to discuss, both from historical issue as well as current snapshot successes and challenges, will be trying to move to a situation where we are applying a variety of advanced molecular and other types of diagnostic approaches, whether next-gen sequencing, transcriptomics, metabolomics, etc., being able to collect, collate, analyze, and use that data to ultimately better guide decision-making for how to treat patients. And as Brian will show imminently, there is a grand vision that many, including Brian and his colleagues, are attempting to enact on, but we still have a lot of way to get there. Certainly there have been a lot of efforts, both in industry, in government, and academia to try and move this forward, and certainly one of the great achievements has been the Foundation One and their next generation, the CDX approach, trying to bring kind of a very broad survey of actionable mutations, as well as things such as MSI and TMB that are important for decision making, at least at this point, for use of immuno-oncology agents. there have been a number of government initiatives and white papers from Institute of Medicine, from NIH and others trying to lay out all the different elements that go into this, from the aspects of making sure that the QC and the validation and ultimately the correlation, the predictive value of these various types of approaches are at the level necessary so that the decisions to do or not do something and what to do are ultimately going to be most appropriate for individual patients. And to that point, it is interesting that, some of these major statements by some of these entities has really attempted to refocus the terminology from what was originally called personalized medicine and what many people still refer to it as to this idea of precision medicine, because it doesn't necessarily mean a unique drug for that unique patient, like a CAR T made from a particular patient and given back to that patient, but the idea of precisely trying to determine what is actionable, what type of drugs are most going to benefit a given patient. So I want to stop there and segue over to Brian to give us a little bit of both kind of the foundational understanding of kind of where we've come from and some of the terminology that we mean here, like this idea of precision medicine and omics and companion diagnostics, as well as, most importantly, to talk about some of the real world day in the life aspects that he and his team at Avera have been experienced by highlighting some actual real world examples of the implementation and the clinical benefits of this type of approach. So
thank you, Jeff. First slide.
Queuing that up, Brian. Away we go.
There we go. So again, good morning all. So basically, the history of cancer treatment mirrors my exact life, which is the chemo was first given in the 40s. When I went through my fellowship at Sloan Kettering, it was not known what caused cancer. Oncogenes were discovered late 70s, early 80s. Bishop and Varmus received the Nobel Prize in, I believe it was 89. And then we progressively began to discover oncogenes and tumor suppressor genes, and the first targeted therapy was approved in 1998. I'm just quoting these from memory. And then the first human genome was sequenced in 2001. This version of Hanahan and Weinberg, in terms of key components of cancer signaling, was published in 2001 and then revised in 2011. And just so that we all have the historical perspective, Foundation Medicine was founded around 2008. So here you have your 6 key signals. These are the original 2001 publication. These are expanded to 12. It used to be thought that every cancer cell possessed each of these key components. In other words, the desire to proliferate, the ability to induce blood vessels, because every cancer needs its own blood vessel supply after it gets beyond 3 millimeters, and then the ability to invade into its current occupying tissue. But in fact, next slide. Next, yeah, perfect. In fact, in about 2015, there was a publication from Nelly Polyak, which showed very clearly that each of these different abilities was held within a particular cell clone. So the cancer is subclonal, and it goes extremely rapidly subclonal. There's been some work from Connie Eves in Vancouver on the West Coast to show that it goes subclonal in 2 weeks. And so, say your gray ones are proliferation, and your green ones are angiogenesis, and your white ones are invasion metastasis. Next. So this is basically what a cancer does. The Harvard people describe this as 4 billion years of evolution held in the life of a single cancer. So it starts with one collection of clones, and as soon as you apply any kind of treatment, then after this, it starts moving in another direction in order to survive. It's basically evolution on steroids. Next. So this is what we show our patients, and they sort of laugh at this. And what we say immediately is, if you imagine you were a terrorist and trying to close down the US airlines, you would have to hit Atlanta in order to close down Delta, Chicago in order to close down American, and also Denver, and also to close down United. And so we say, well, what we are trying to do is close down the 3 critical hubs in your tumor. So for instance, on the right-hand side, upper right, is DNA repair. Right in the middle on the left is the whole PI3 kinase, AKT, mTOR, and beneath that is the RAS-RAF, MEK, ERK. Next. So the other thing which is utterly vital is measuring tumoral circulating DNA. So every time you or I wash my hands in the sink, we leave some dead skin cells behind, and indeed the normal skin is shedding little bits of its own DNA all the time, which are the little brown parts. And the tumor is equally shedding little bits of its DNA all the time, which are the blue bits of DNA in the bloodstream. And so by constantly monitoring the cell-free DNA, you will indeed pick up any alterations in that genome. So the appearance of the red little clone that I showed in the last slide, you will pick that up by constantly monitoring the cell-free DNA. So we regard monitoring every 3 months of the cell-free DNA as ideal. Next. So this is a Charlie Swanton slide showing something like 9 biopsies, which were all different metastatic sites in a kidney, and showing that if you only biopsy one of these, then you basically pick up the red things on the extreme left-hand side. But by following cell-free DNA, because the blood is going through all 9 of these, then you pick up the entire spectrum. The only problem at the moment is I believe the Foundation one is still monitoring at 1200-fold for its Foundation CDX, which is a tissue test, whereas in their Foundation liquid, they are monitoring at 5000-fold. You probably need greater depth and a greater number of genes. Next. So these are the cancer treatment eras, and I've been through them. Chemo in the 40s, non-specific therapies, targeted therapies coming in 98, genome area coming in 2001, and immunotherapy coming in 2011. Next. So there is an increasing shift. There are something like 95 matching targeted therapies approved by the FDA. We've moved from the very crude chemotherapy into these highly targeted therapies matching specific mutations or amplifications in the genome. And so we're moving progressively to a more targeted world. Next. So progressively, this is a very old Janet Dancy publication in Cell. You can see all the alterations on the left, all the matching drugs on the right. And as I said, FDA has now approved something like 95, and their speed of approving these drugs is accelerating. Next. So we're beginning less to define a tumor by its site of origin, although that is critically important. Foundation Medicine sequences or shows you 400 targeted genes, which are all involved in cancer. And indeed, the most important thing for treating each patient's tumor is which of, I mean, this one shows just like 8 on the top. In fact, which of these 400 genes have mutated, and to be able to knock down all of the clonal populations. Next. So this just shows the sheer complexity. If you're looking at BRCA1 and BRCA2 and PALB2, these show the individual mutations, and very often you need a specific drug for each of these mutations. Next. So what we believe is that sequencing a diagnosis not sequencing a diagnosis will basically become malpractice within 5 years. Your probability of response by line of therapy is shown here. It's the old Vince DeVita slide. And by the way, I highly recommend his wonderful book, The Death of Cancer. So as you can see, and as you know from your own practice, the most aggressive therapy should be given at diagnosis, right at the lead. And so therefore we believe that sequencing as soon as the tumor appears and treating right at that point and giving matched therapies for each of the clones is going to be the way to go. Next. The slide changes are a bit slow, Jeff. I'm not sure why. So this is a typical patient that we have. This is a 37-year-old who has had stage 4 metastatic breast cancer. She'd been given absolutely the best treatment, including the NSABP-B47 protocol at diagnosis. But in spite of that, she had huge local recurrence, but it was literally stage 4 on the left chest in 2015. And so we sequenced her. Let me just show you the next slide. This, thank you, Jeff. This is the key result of her sequencing. She had a PIK3CA, which matched everolimus. She had a CCND1, which matched Palbo. She'd had her ovaries out, so the disease was driven by estrogen, so we gave her an aromatase inhibitor. And she'd also got a couple of other alterations in FGF4 and 19 and PALB2. So we started with a combination of an AI, Palbo, and Evero. Next. So this shows one of the more common things now. If you actually look on the right of the slide, you will see that CCND1, FGF4, and 19 all occurring on 11q, all in the same spot. And we're finding this more and more, that several of the changes are chromosomally located in the same position. Next. So I've already been through this. We treated her with LET, Palbo, Evero. This showed a absolutely complete response. All disease disappeared. Next. So we did follow her with, in this case, Guardant, and this basically shows the appearance of an RB mutant clone, which is a resistance mechanism to the Palbo. So this is literally showing the tumor in evolution responding to the Palbo therapy. Next. There's the RB mutation right on the top line, and you can see the increase in this as a result of the Palbo treatment. Next. So we stopped the Palbo, and we then flipped, changed the Palbo basically to pazopanib. She remained in complete response on that. But she didn't like the drug very much. Next. So just to finish that story off, we flipped the pazopanib to, I think it was olaparib for the PAL-B2, and she's remained in complete response all the way through this. So if you notice, she's maintained complete response, and we changed the treatment twice based upon the circulating tumoral DNA and the inflection points in the tumor. And we feel that that is going to be the way the practice is changed in the future. This is a second patient I'll do extremely quickly. Very advanced bone, liver, multiple other places. It was a stage 4 pancreatocholangiocarcinoma. This chap was only 38 when we started treating him. Next. So this may sound very strange, but he was very fortunate to have Lynch syndrome and MLH1, so we could give him pembro. He had a RET mutation, which matched cabozantinib perfectly, and then we gave him baseline cisplatin. Next, and progressively, he had a complete response. This just shows basically the treatments. We dose the drugs down so that we can do these N of 1 combinations. Each therapy, each multiple combination is matched to the exact genomics that that patient has. Next. This should show, yes, this should show his circulating tumoral DNA. This is again the Garden 360 report and shows all of the clones completely disappearing on this triple therapy. Next. And this just shows everything. So thank you. So this is our best outcome. We've got over 55% having a PR or CR RECIST defined. We've got more than 90% of patients under control. Next. These are our neoadjuvant patients. We treated something like 75, which we're now writing up, had exceptional results. Next. These are our, next. So these are our metastatic patients. So in brief, this is an average of 4th line therapy. So they've already had 3 treatments in the metastatic setting. And we managed an average of 55% RECIST responses in the left column, 20 plus 35, in these stage 4 metastatic breast patients. Next. These are our GYN results. These are absolutely remarkable. We have 41 plus 35 equals 76% response rate in our ovarian population. Next. And these are the most common combinations that we've used. This shows you just the example. We've lowered dose of each of these, and each of these are given on a customized basis. And Jeff, I believe that that is slide 30.
All right. Thank you very much, Brian.
My pleasure.
And of course, you know, as we start the dialogue and the fireside chat proper now, if anyone has any questions, either to the slides that Brian has just presented or as we're having our discussion, please send them through the system and we will attempt to work them in. And if for some reason we don't get to any particular question, we will do our best to follow up subsequent to the webinar. So let's start at kind of the highest level here, Brian, and that is, you know, with these phenomenal examples that you've shown, maybe you can speak to some of the advances that you've made, as well as some of the challenges in really reducing this to practice, right? There is being able to get access to these drugs. Some of these drugs may or may not be formally approved in the indications for which you want to use them. That then brings in issues of the access and, of course, then reimbursement. And I'm sure, you know, many of the audience members are saying, well, how can this happen? how can you do this? What does it take to do it? And then how can we broaden it from, okay, Brian's a special case. He's the pinnacle of his field. He's at a great institution that is allowing him to do that. So that's really kind of two-part question. First, kind of how have you been able to enact this? What challenges have you had to overcome? What remain? And then how can we see this kind of broadening out to more and more centers and ultimately to a broader swath of treatment centers?
So, Jeff, you're absolutely bang on target. A lot of people have asked after being in Sloan Kettering and Emory and McGill, what the hell I'm doing in Sioux Falls, South Dakota. So I moved here for $100 million, which was available for breast cancer. I flipped institutions since, but both institutions in Sioux Falls have given us a great deal of money in order to conduct these sequencing matched therapies. So we've have we changed slides or?
Yeah, I just went back to a general slide. I can go back to yours if you want.
No, it's fine. It's fine. So basically, you know, the big plus here is, as Roselle Kurzrock always says, we are in the Wild West and we're not as restricted in treatment. We have money and we're not restricted by NCCN guidelines quite as much. We do follow NCCN guidelines, but we expand upon them. And so the big thing is we have a team. So we have a sequencing team that does all the interpretation of the data. I'm sorry, we have a signaling team that does all the interpretation of the data. We have a sequencing team that does RNA-seq and some proteomics, etc. We've got 2 patient advocates. We have 5 pharmacists that are specially trained in doing these N of 1 combinations. We have nurse practitioners that know the processes of ordering the Foundation 1, the Foundation acts cold, and we have an excellent relationship with both Guardant and Foundation 1, so that we're able to access the platforms. Nevertheless, the challenges, just as you said, strongly remain. Access to the drugs, trying to do repeated liquid biopsies. You know, people are just not into that sense anymore of doing 3 liquid biopsies in a row, which is at least what I love. If this were my own life, I would be doing these every 3 months as long as I possibly could. I mean, these liquid biopsies can be fantastic. You can have a patient on Herceptin for 3-4 years and then take a liquid biopsy. And then if it's negative, you can stop. If it's positive, then you can look at the results of that liquid biopsy and treat accordingly, either continue with the Herceptin or change to something else. But the biggest problem is access to repeated testing, access to drugs, having an adequately built team of your own specialists so that you can actually practice this.
That's great, Brian. Well, let's talk a little bit about this the biopsy aspect, because whether it's serial standard biopsies or liquid biopsies, looking at CTCs or circulating DNA, I think first of all, not everyone listening in may know kind of what the current state of these are and to what degree we are kind of state of the art, particularly in what we can identify from the circulating DNA. Maybe you can talk a little about some of the technologies and if in particular there's kind of anything remaining there to improve upon, either at the technological level or otherwise.
Well, there are always things to be improved upon at the technological level, Jeff. I'm going to keep my answer relatively brief. So diagnosing at first-line metastatic disease is pretty much approved by all NCCN guidelines and covered by insurance, and that I would encourage everyone to do and sequence. We've been able to pretty much, so the tissue biopsy That's the Foundation 1 test, and you can usually get that done without any hassle. And if there are problems, then you speak to Foundation 1 personally, and generally they will cover it. Sometimes under trials and other circumstances, we can do a repeat biopsy at 3 weeks or 3 months. I-SPY, for instance, the I-SPY trial does a routine second biopsy at 3 weeks, which is perfect. But you need that second biopsy has to be paid for one way or another. And at the moment, I'm raising funds in order to build a community oncology network so that those funds are available for a second biopsy. Doing the foundation acts repeatedly really does take a team. Although I have several of my fellows and colleagues that do manage to do the foundation acts, at least do a couple of them and get them reimbursed one way or the other. Getting the matching drugs is tougher. It's you really do need a patient advocate. So in my building this community oncology network with several colleagues, one of the things we're going to do is absolutely ensure that funding is available for a patient advocate to serve each center.
Great. I was glad to hear that you mentioned about raising funds to kind of broaden this out into the community. Could you talk a little about kind of what other centers are kind of replicating this Avera model? Are they doing it in any way differently, or are you all kind of more or less kind of harmonizing yourselves and learning from each other? And then, so that's the first question. I have a follow-on after that.
So there's one other center that's practicing exactly in this manner, and that is Rizal Kurzrock at UCSD and her entire team. So we have just done a joint study called iPredict, and that has either just been published or currently being published in Nature Medicine. So that is the closest. We're hoping that the other people will join this kind of network. And that was, in fact, funded by Foundation 1.
So 2 centers currently performing this state-of-the-art approach with the phenomenal outcomes you're showing with basically appropriate utilization of drugs at hand, not even talking about deploying, novel next generation targets or modalities, etc.
Correct.
Why is that?
Let me just add.
Yeah, go ahead.
I'm sorry?
No, go ahead.
Let me just
Brian.
add one more point, which is that the WIN consortium based out of Paris has 41 international sites across 21 countries. And that is absolutely committed to doing triple therapy and works extremely closely with the FDA. So at the moment, we have our first 3-drug combination from Pfizer for lung cancer, and we are one of 6 phase 1 sites which are, just testing this triple combination. What we're hoping for, obviously, is to get multiple pharmaceutical companies involved. And there are 2 aspects to this. One is to have basically a compendium of 15 combinations from different companies. And then we have also built what is known as a SIMS algorithm. This is a matching transcriptomics algorithm, which will point each patient to one of those 15 combinations.
Right. So this WIN consortium, Paris, you said they're working with the FDA, but that still begs the question that I was going to ask is, there's you, there's Rizal at UCSD, my undergraduate alma mater, but
yep.
Why aren't there more here in the U.S.? What's the limiting factor for this right now? Certainly, there are many other centers that are making noise about their doing precision medicine in some way, shape, or form, or certainly have a lot of either clinical or translational research in that regard. What is the limitation to there being more such centers than these two? Is it simply funding? Is it, maybe you can speak to that.
No problem. So the dominant one is that NCCN guidelines are still absolutely dominant, and they have not yet flexed to the point of allowing N-of-1 therapy. What we're trying to do now with the publication in Nature Medicine, unlike Wynn is doing, Wynn also has a publication coming out of its original, what they call winter trial in Nature Medicine. What we're trying to do is build up a body of evidence that will convince pharma, FDA, and all of the testing companies of the importance of this N-of-1 approach. The other limitation on this is that there are various basket trials available now, like NCI Match and Taper. The originators of these trials are my very, very closest friends, but the limitation is they are using one drug at a time. So with cancer being subclonal in populations, you treat with one drug, matching one mutation or amplification, and then you eventually fail, and then you move on to the next line of therapy. These single basket therapies are not curative.
Yeah. So actually, I'm going to take a break from my list of questions and actually attempt to address, because a bunch of questions have come in. So let's take a look at some of these here. First one is a very, very specific question about what about the role or importance of Esr1 mutations in metastatic breast cancer. You can comment on that at all in the context of what you're doing, or?
Yeah, I mean, so ESR1 mutations, you barely ever see them early on in the disease. They change from something like 1% to 20% in the metastatic disease. They are usually felt to be a result of aromatase inhibitor therapy, although we've had one or two just appearing on tamoxifen, and we're treating these at the moment with fulvestrant, which is not very potent. It's not great. There are various SERMs and SERDs which are coming out in the pharmaceutical industry now, which will treat these ESR1 mutations much better.
Right. All right. That's the most kind of very narrow, specific question. Now there are a couple that touch or amplify on some of the things that we've been talking about or that I was asking about. So one is, I think, an interesting one. So, and that is, and you did refer to kind of the down dosing, but the question is about, like, how do you determine the appropriate dose for each of these individual products when you're kind of putting them together to match a particular patient? So can you talk a little about that, in terms of the kind of the labeled dose versus, how you're determining what to do when you're putting 2 or 3 or more together?
So this has been a path of much anxiety. So we've dosed down right from the beginning for these triple combinations, and it's been somewhat trial and error. We've never had one patient hospitalized from overdosing, which is utterly remarkable. Say everolimus is normally given at 10 milligrams as per the package insert. We give it at 2.5 in combination. Palbociclib, we never give more than 75, although the label is 125. Crizotinib is generally given at 800, and we give that down to literally 200 milligrams in these triple combinations. Something like olaparib, we generally dose at 200. If we're adding chemo in, we will often do carboplatin with an AUC of 1.5. We just because of our superb signaling and pharmacy team, we've just managed to hit on the right combinations, the absolute perfect doses for these combinations.
So, at least for the moment, would you say that there's as much art as science to it? But obviously, as you're doing this, you're building up a data set, which hopefully would be informative going forward, not only for you, but potentially broadly for others in the field.
Jeff, you have it exactly. And we're publishing. We're about to publish some of this stuff, so it'll be out there within this year.
Great, great. What about, there's a question about kind of the temporal aspects of these genetic mutations, not just the spatial ones. Can you talk a little about that?
What do you mean by the spatial ones?
Spatial in terms of different sites in the tumor, the primary of the metastatic site, but also the idea of temporal over time, how these change. You talked about, the serial sampling every 3 months. Can you talk a little about what you're finding when you're doing that? I mean, is it the kind of the usual suspects of obvious resistance pathways that you're seeing? And does that tell you anything about, do you have to be kind of, by necessity, do you need to be sequential? And I know we've talked about this before, or once there's a database of, 80% of the time we see this pathway being kicked in sequentially, then would you want to attempt to load that up front with the hit heavy idea that you said before, or would you always need to wait? And I know part of that's about the limit of how many drugs you could put together in terms of tolerability, but.
So let me respond by just saying, obviously that slide that I showed of Charlie Swanton's was an autopsy where you see the nine different sites within the kidney, and they sequenced it on the autopsy each of those sites. So what we do as a routine is that we sequence right at the beginning the tumor, and then we treat it. We try to do a foundation act at the same time to see the level of correlation, and then we follow on liquid biopsies. And we believe that liquid biopsies are going to become more and more important as we move forwards. I do think, yes, that if you take a classical situation like ALK and the resistance paths, that it develops, EGFR and the resistance path that it develops, some of these are becoming more and more predictable, and we will progressively, in my humble opinion, although this is not shared by everyone, but in my humble opinion, we'll be treating right up front with, say, a first and a third generation ALK inhibitor right up front in anticipation of these resistance pathways.
Great, good. So another question related to this idea of the liquid biopsies, which is specifically, how available, how accurate, how precise are these types of tests?
I think they're becoming more precise by the day. They're becoming more fine-tuned. We used to see overreads of amplifications, etc. I think they're becoming much better. If you take people like Grail, they're not sequencing at 5,000-fold, they're sequencing at 60,000-fold, so you get huge depth. And I think we're only at the beginning in terms of doing DNA. So, I mean, if you just compare your body to a house, DNA is basically the architect. If you look at RNA, DNA is the architect, RNA is basically the construction crew, and then the proteins, which actually run everything, they're the final product. So we're progressively moving up DNA. Many people now are doing RNA-seq in order to cover the transcriptome, and I think soon, I know that one company at least is going to be bringing back its protein assays where they're doing 30, 35 ELISAs of phosphoproteins, I think we're progressively going to move up the ladder of DNA to RNA to protein.
Great. So that's so. Now, and when you talk about transcriptomics, I mean, the majority of that, though, is focused on transcripts that are ultimately, expressed as proteins. What about many of the regulatory and, long non-coding sequences and microRNAs and a lot of the other interesting and constantly being discovered aspects of RNA that are important in the control and dysregulation in cancer?
So you're absolutely right. So let me make one point first, which is Andrea Califano at Columbia University is now using his New York-based CLEAR transcriptomics, RNA-seq, to come up with predictors of what he feels are the phosphoproteomic drivers. He's looking for those signatures in the transcriptome. And then next one, I'm sorry, I'm losing track on the questions, but yes, the regulatory elements. So this is going to be vast and expanding over the next 10 years. At the moment, you have something like 1.5% of your genome expressed. 98.5% of this is regulatory areas, and progressively people are going to move on whole genome sequencing and look at the regulatory regions and determine how you can treat the regulators as well as the expressed genes.
Great. Yeah, and of course, that's some of the leading edge of very early types of novel therapeutic modalities that are either exploit or harness those things like microRNAs or are targeting them or increasingly targeting transcription factors, sometimes through more novel approaches like PROTACs, et cetera. So a lot of cool stuff really will become available to be used once that information is being generated as to the relevance of those elements to the patient's cancer.
Correct. So I mean,
so since we're on this topic, one of the questions I had also related was, what about exosomes? A lot of people are analyzing them, looking at them. It's a fascinating idea that these things get exported and somehow either modulate the tumor microenvironment or, in fact, kind of help seed the metastatic niche. To what degree is that also going to need to be on this radar screen? And I know we're kind of expanding this to all sorts of future complexity when there's a lot still to accomplish at the most basic level of getting patients access to these triple agents, but trying to be a little forward-looking here.
I think you're going to see an explosion in the next 5 years of one, looking at regulatory areas, number two, looking at things like exosomes, number three, looking at things like mitochondrial DNA. There's going to be much more of a focus on that. If you look at the work that Joe Gray and Mina Bissell are doing, they're basically looking at 3D vision of the tumor. So you can see all of the tumor, the surrounding stroma, where exactly the T cells are, macrophages, all of the components of the stroma and their cellular proximity. There's going to be a lot more on the exact architecture of the tumor, which is going to be part of literally the diagnostic spectrum. I think we're just going to see an explosion of the next year, including much more transcriptomics, introduction of proteomics, etc.
Yeah, and I'm glad you mentioned the idea of kind of the broadening, kind of the spatial analyses. I saw a great talk that Jerome Gallon did at AACR and during one of the educational sessions, referring to a key Cell paper that they had published, kind of tracking the interaction of the tumor and the host immunity, both in space and in time, showing the critical role of the immune system playing in this Darwinian evolution of the tumor that you referred to earlier in your slides.
Absolutely.
Yeah. And it also, you mentioned some of the approaches, had an interesting discussion with people we know at NanoString talking about their geomics spatial profiling system too, which is, again, very cool. And all these approaches that allows one to better understand. I mean, it's one thing to keep talking about the increasingly micro level of transcriptomics or metabolomics or regulatory elements, et cetera, but we shouldn't forget the importance still of the kind of the more macro view of the interaction of these elements in that kind of tumor immune space and the immune synapse.
Absolutely, Jeff.
So speaking of that, then let's talk about, so in the slides that you referred to, clearly the focus right now, because, I use the term low-hanging fruit only in quotes because it's still obviously not easy to do, but we're talking about mostly actionable mutations. There are clearly kind of higher level types of analyses that maybe suggest that, certain drugs, aren't going to need to be used because there's a specific mutation, but maybe there's some type of dysregulation that necessitates their being used, whether it's amplification or something. And so the idea of not just these individual changes, but the idea of looking at higher level complexity, signatures, et cetera. Any comments about that?
Well, tumors very often split into those that are predominantly mutated and those that are predominantly driven by amplification. So we do see both sides. I don't know what else to say. I mean, we've done extremely well with matching at the DNA level. We could do far better. We've had some patients who've failed all matches. So obviously we're not doing a perfect job. On the other hand, I will say that we've got some breast cancer patients who are out literally 14th and 15th line and doing incredibly well. So we had one breast cancer patient whose tumor was driven by HER2. She was on 13th line HER2 therapy and failing that altogether, progressing. We sequenced her because she was progressing. We did a biopsy. We did a perfect matched combination to that, and she progressed right through it. And then we went back to HER2 therapy, and she had a complete response. So it just goes to show the rapidity with which the tumor will flex. It will go back and forth, and if you confuse it by giving a different therapy, even though the different therapy doesn't work, and then you go back to your original, that will often work. This is just like the childhood leukemias where you could use adriamycin 6 or 8 times, providing you had drugs in between.
Right. What about, so because, there's still a lot of interest, some would say frenzy, around IO, and some of at least one of your studies did refer to use of the checkpoint inhibitors, but can you speak a little bit about kind of how you're thinking about, better deploying those, given that right now we're basically either have PDL levels or TMB or, MSI, but clearly there's, it's somewhat in its infancy, if you will, for how best to deploy these, especially how to think about, those patients who have innate or ultimately acquired resistance to the checkpoint. So some commentary there?
So, Jeff, I don't have much to add on this at the moment. I mean, you know that we are having a panel on this at Defined Health on GYN tumors. One of the experts in the field commented how PD-L1 and TMB do not appear to correlate so well with IO success in GYN malignancies. I think we are, just as you said, right at the infancy of these platforms, and I think as we move up to 30, 40, 50 IO drugs over the next 5 years, as we move into the T-cell stimulators, etc., I do immune cell activators, except multiple things, macrophage, inhibitors, I think we are going to find a whole different set of immune biomarkers which will correlate better. I do still think that these markers are probably going to have some disease specificity to them.
Great. Okay. There's one more question from the audience, and then I have one more question, and I think that will start to round out our fireside chat. So the question is, are you looking at any types of preemptive or prevention approaches for higher risk people, which is a great question because the opening panel at Cancer Progress will be about kind of chemo and now immunoprevention and some of the struggles historically of getting the industry interested in doing that, and how a number of things have started to change both in academia, industry, and the drugs at hand to perhaps better enable the idea of interceding early, either earlier in line or earlier in stage of disease. So can you talk about that a bit?
So I will make 3 comments on this. Yeah, and I was right at the beginning of the development of monoclonal antibodies. And after about 18 years, people knew that antibodies would never, ever, ever work. And then slowly the first antibody results came out. Herceptin was one of the early ones, etc. And so finally, it took a long time to show that monoclonal antibodies did, in fact, were excellent therapeutic moieties. Needed a lot of tweaking. TDM1 took, the whole idea of having a poison attached to a HER2 ligand or whatever, that took many, many years. I think a lot of people are disappointed in the overall prevention field. There have been multiple attempts, most of which have been negative. I think the tide is about to turn in prevention. And I do think that one of the key aspects are things like what Grail is trying to do, what Hopkins is trying to do, which is incorporate very high-depth sequencing or a combination of genes and proteins. Hopkins does something like 25. If I remember correctly, it's something like 12 genes, 9 proteins, something like this. And incorporating that into an early cancer detection test, which would basically be incorporated into your annual physical. So you would sequence at very high depth using a very select number of genes or proteins or RNAs or alterations, and that would predict when you have a cancer. And if such a test was successful, and if you could get the regulatory approval, obviously it would need a trial, you'd be able to treat cancer at its very earliest stage, and literally cancer, metastatic disease would disappear off the planet.
Yeah, I'm going to play devil's advocate a bit on that. I mean, that's, I think we are getting better tools. Part of this may be enabled by, or certainly some of the idea around the use of immuno-oncology agents, given, I don't know if I want to say safer profile, certainly a different profile than other agents. And so there are a lot of efforts moving those into adjuvant, et cetera, and into earlier stage disease. But there certainly are many studies that show that even at the time of primary, even if it's in the earliest stages of primary, there may well already be, metastatic cells, already out there, so that we're already from almost day one in a subclonal type of situation. Any thoughts on that, or?
There's no doubt that the tumor is subclonal and metastatic at that point. But having said that, you can treat before those metastatic tumors are radiologically evident. You can detect the tumor by some kind of sequencing at a very early stage before you can pick anything up radiologically.
Yep. All right, and then I just have one last question. So go ahead.
There's one other comment I just want to make on that. There is no question that some form of immunotherapy will be incorporated in the treatment at the earliest stage.
Yeah, and we'll certainly have a number of different discussions across various panels at Cancer Progress on that, I have no doubt. So I have one last question, and that is, we've talked a lot, most importantly, of course, about, the application to patients, but in order to get to patients, or to better get more drugs to patients, the application of these type of precision medicine to clinical trials and even early drug discovery. Can you talk a little about kind of what you and colleagues and others are doing in that regard to try and better enable, bringing individual as well as collections of drugs, new drugs, novel drugs, individually and in combination together, so that one is almost more at the outset having this precision medicine concept in mind?
So there is one, the answer to this is one single word, which is money. We have in fact written such an N-of-1 trial already, but in order to do this, you need to pay for extra biopsies. You need to pay for both tissue biopsies and liquid biopsies. You need to have extra money for that. You need to pay for medication acquisition specialists in order to access the drugs. And you need nurse practitioners, etc., who are incredibly well versed in the practice in the local hospital, as well as a central signaling and sequencing team that has brilliant expertise. So this takes a significant amount of money. We're beginning with one protocol, N-of-1 protocol, and 4 or 5 centers. I think one of the big pluses, which is coming now, is Skip Burris is just about to become the next ASCO president, and he is going to push community healthcare, which is where 85% of cancer therapeutics are delivered. He's going to push access to those community centers as part of his ASCO mandate. So I think that the future is very optimistic. Whenever, all I can say to the audience and to everyone else listening in is that please, when you see these fundraising aspects going towards N-of-1, not for profit, please, give to them, support them, help us in this mission.
All right, great. I think I'm good. There are no more audience questions. Any parting comments from you, Brian?
No, it's been great. I'm delighted the audience attended this. And as an outsider, although I will be there, I encourage everyone to attend Jeff's Defined Health Conference in New York, which I think is what, in 3 weeks, 4 weeks?
Yeah, it's sooner than we'd like, but yes, in May 7th and 8th.
There we go.
All right, thank you. Thomas, you want to close us out?
Sure. I want to thank everyone. The presentation and Q&A have been terrific. Again, this webcast will be posted and links distributed by early next week. And I'd say with that, we thank Brian and Jeff for this terrific session and for your attendance and discussion, and wish you all a good day and a great weekend. Thank you.
Thank you, Thomas and the Kinexum team. Thank you, Defined Health folks, and most especially, thank you, Brian and the team you work with, and of course the patients who are always front and center.
The patients are always front and center. Thank you, Jeff and Thomas.
Great, thank you. Take care, everyone. Have a good weekend.
Great, bye.
Bye-bye.